So, I am very excited to present to you Lindsey Backman, a fellow Floridian who grew up in Tampa, Florida. She studied chemistry at University of Florida and is currently a graduate student in Catherine Drennan's lab in Building 68, also known as the Haven of Labradoodles. And if you don't know, now you know. When asked what's the coolest biological phenomenon she's ever witnessed in person, Lindsey replied it was hearing crickets starting to chirp midday in Tennessee when the solar eclipse was happening in 2017 and near totality, which, for the record, that's really cool. I'm jealous. And so, I'd like to welcome Lindsey up here to tell us all about boop.
So I truly never imagined, when starting my PhD, that part of my thesis would be focused on understanding the chemistry of why poop smells bad. But that is exactly what I've been working on lately, and it is an unbelievably fascinating topic. So, in all honesty, we accidentally stepped into this project. Our underlying fascination lies in the human gut microbiome. We like to think of ourselves as autonomous human beings, but in reality, the bacteria that inhabit us are performing a wide range of chemical reactions that impact mammalian health. The large molecules that actually perform these chemical reactions are called enzymes. And in the Drennan lab, we are interested in identifying new enzymes, currently of unknown function, who are abundant in the human gut microbiome. And that is exactly how we discovered IAD, this enzyme that makes skatal, a molecule that is extremely odorous and is the reason why poop smells bad. So you might be wondering, why does this even matter? And I had the exact same question at first, and I was surprised to learn that the agricultural industry has been searching for this enzyme for a long time. Why? Three main reasons. First of all, zoning regulations. So there are currently very strict zoning regulations of how far communities must be from farms based off of smell. Secondly, bore taint. So skatal, the degradation of skatal in the liver is actually inhibited by male hormones. And so skatal can actually accumulate in boar tissue, thus making perfectly good meat smell tainted or spoiled. And lastly, skatal is actually toxic to lung cells in cattle, thus causing fatal diseases in these animals. So if we could find a way to inhibit IAD, the enzyme that makes skatal, we could potentially address these issues. So in order to do this, we first need to understand how IAD functions in the first place. And in the Drennan lab, one of our favorite ways to do this is a technique called X-ray crystallography. So with X-ray crystallography, we can basically take snapshots of our enzyme. We can get pictures before it does its chemistry, while it's in action, and after it's made skatal. And we can start to understand how this enzyme works and how we could potentially stop it from working. So as of now, I have recently solved this initial structure of IAD, and we're currently working to get a higher resolution structure and to eventually explore the option of designing inhibitors for IAD. Overall, this has been an incredibly exciting project to work on, and hopefully with this work, we can help make the world stink just a little less.